somebody just hit the button. Okay, probably Rebecca. Thank you. So yeah, we can get started. I'll just very briefly introduce you. So Dominique, we're very happy to have you coming to us from Paris this morning. And for those who don't already know, which is probably nobody on the Dewpoint team, because we've already been talking about you, your work in P-bodies especially, but more broadly, the role of RNA in condensates is something that we've been watching and are very excited in hearing about. And today you'll be talking about mRNA storage in P-bodies. And you've also, Dominique has also published on some helicases, which are also topics that come up pretty frequently these days in the condensate community. So that might be another topic of a discussion as well. So we thank you again for giving this lecture, and you can take it away.
Okay, thank you for the invitation, particularly in this very special moment. So I'm going to share my screen. Okay, just can you confirm you see it? No. Can you confirm you see my screen or?
Yes. Yep. Okay, we can see your screen. Yep, we can see your screen now.
Okay, that's fine. So my lab is located in Paris, so close to the Seine. It's in the Institute of Biology Paris-Seine. So I would be here, my lab would be here. So today I'm going to talk about the GC content that shapes mRNA storage in human P-bodies, or rather how we came to that idea and to have evidence for that. So in general, so you are a particular audience, so you know about all that, but post-transcriptional regulations of mRNA, it's a story of RNA binding proteins that are going to control if these RNA are going to be translated, stored without translation, or decayed in the cytoplasm. But it's also a story of cytoplasmic RNP granule, where the RNA and the proteins responsible for this regulation are accumulating. And we are particularly interested in this aspect post-transcriptional regulations. In general, these granules, these RNP granules, they have very different names: germ granules, neuronal granules, P-bodies, trace granules, now even translation factories, depending on the cell type where they have been found, like in germ cells or in neurons or in all cell types for P-bodies. But it's not only a matter of name or of cell lines, of cell type. It's also, these granules, they also differ by their composition, their morphology, and maybe their function. But globally, overall, the questions, at least the way we see these granules, the questions are more or less the same for all of them. How do they assemble? What do they contain in terms of proteins and RNAs? What is a function in RNA metabolism? And further than that, what is a function in cell physiology? So in our case, in the recent years at least, we have been particularly interested in P-bodies in human cells. So these P-bodies, they are here in an epithelial cell, human cell. These red dots, red because they are labeled with the expression of a DDX6 helicase fused to RFP. In the absence of this protein, they would not be visible, in contrast to these other gray granules here, which are visible in phase contrast because they are surrounded by membranes. These P-bodies, they have been observed in animals, in vegetables, from yeast to human. Since the beginning, we know that they contain a set of proteins related to mRNA metabolism function, mRNA decapping, mRNA decay, specialized pathways for mRNA decay, such as NMD, nonsense-mediated decay, RNA interference proteins, which act both on mRNA decay and mRNA translation, and factors involved in translation repression. So from this first list of components, everyone postulated that they should be important for mRNA decay, translation regulation, and RNA interference. However, how they do, what do they function within this different, with respect to this different regulation pathway, has been a mystery, and is probably still a mystery. So at least we know that they can translate these PBDs. This has been, first of all, nobody has seen any signal using antibodies directed against ribosomal proteins. But then we also confirmed that by combining electron microscopy with in situ hybridization using 28S probes or 18S probes. So here we have this DDX6 helicase I was talking about, fused to RFP. This time it is the endogenous DDX6 protein. And the antibody is coupled to large gold particles, 15 nanometer diameter. So we know that this is a Peabody. And the in situ, the 28S probe or 18S probes are fused, coupled to small gold particle, 5 nanometers. And as you see, all the small particles are surrounding, are in the surrounding cytosol, but they are not located in Peabodys. It's true for the two ribosomal probes. And when we quantified that, we found that indeed in granules, the granules are very devoid of 28S and 18S signal compared to the surrounding cytosol. Some groups, or for some other granules, have proposed that maybe they would be a crown of ribosomes at the surface of the granules. We do not see that at all. In the periphery, the concentration, the density of gold particles is very similar to in the surrounding cytosol. This is very much in contrast with stress granules, another type of granules that which is induced by stress and contains also untranslated mRNA. The same approach that we did a few years ago led to the evidence of accumulation of 18S ribosomal RNA, but not 28S, confirming things with since observation by immunofluorescence that the stress granules contain the small ribosomal subunit, but not the large ones. So just as to compare these two ones, which form in the same cell type, both granules contain silent RNAs, but the Peabody RNA are totally out of translation, while the stress granules contain mRNA, which are pre-initiated for translation. Just to extend a little bit this comparison between Peabodys and stress granules, it has been observed by many labs, including our, that the Peabodys here in green are very often found in close contact to stress granules when the cells are stressed, here in red. We also visualize that in electron microscopy. Here is a stress granule, and here the neighboring Peabody. Nevertheless, the two types of granules, we never found them intermingling, and in fact they have different structure. Stress granules have granular ultrastructure, probably related to the presence of the small ribosomal subunit, while Peabodys, it is enlarged here, they have a fibrillar ultrastructure. We also found that these contacts, they do not form after the two types of granule form, but in fact, many stress granules, not all of them, but the majority of them assemble at the contact of pre-existing Peabodys. We don't know why. It's like a seeding mechanism that will be worth to study, but so far we have no clue of what is the origin of this. Now, to come back to Peabodys, as you know, they are formed following liquid-liquid phase separation. So in electron microscopy, they clearly have no membranes. Here it is the endogenous DDX6 protein, which is detected through immunostaining. We have conducted several studies showing that 3 proteins are required in human cells for Peabody assembly. It's this DDX6 helicase, the dead box family, 4ET and LSM14A, which are RNA binding proteins and also which interact together and with DDX6. We don't know how they interplay together, but we know that the 3 of them, if you silence even one of the three, Peabody's disappear and cannot be reassembled by any trigger we tested so far. So we got particularly interested in this DDX6 helicase for several reasons, but maybe mainly because it's a helicase which is very highly expressed in human cells. Concentration, we measure that the concentration of the protein is of the order of 3.3 micromolar in the cells. And by counting the gold particle in this immunostaining, we could calculate that the concentration inside the Peabody's reaches 0.5 millimolar, which is extremely high for a protein. We don't know if that would enhance the enzymatic activity of the DDX6, or maybe contact with some of its protein partners, or maybe this very high concentration would do the opposite, like decrease the activity of the protein because just it's too viscous. But it should somehow change the property of the complex, the activity of the complexes, which are contained in Peabody's. Okay. One of the reasons it was very difficult to find a function for Peabody's is that by silencing any of these proteins, we can make disappear the Peabody, dissolve the Peabody's, but that doesn't lead to measure defects, visible defects in the cells. Everything in terms of translation, in terms of decay, in terms of RNA interference continues to happen. And it has been difficult to prove that these Peabody's were required by any way. One missing information probably to understand the function of Peabody was which RNA are in there. In fact, maybe there would be naturally only a few RNA that would accumulate there, explaining why when we suppress the Peabody, we don't see major defects, and we would need to imagine some particular test to evidence the function of Peabody's. So to know which RNA are in Peabody's, the only way probably to make it systematically is to purify the Peabody's. This has been a major challenge for all labs which tried. First reason being that nobody could find any protocol of differential centrifugation that would be efficient at purifying Peabody's without co-purifying membranous organelles. So mitochondria in particular come with, but many other organelles, all these membranous organelles here of the same size. Another challenge is that they are very small in size. There is a low number per cell. They have no membranes. But maybe, at least to my opinion, one of the major difficulty is how much of any marker we have is really in Peabody's. So take this DDX6 protein, it's really hugely concentrated in Peabody's. But nevertheless, if you calculate how many molecules are in Peabody's versus outside of Peabody's, you find that not more than 20% of total DDX6 is located in Peabody's at any time, despite the fact that it is 170-fold concentrated in Peabody's. But they are too small and very scarce. So it's probably true for any type of, any Peabody marker. So we have to consider that Peabody's contain a high concentration of all the protein that will be found in it, but these proteins, they also exist outside. So any approach using immunoprecipitation is going to co-precipitate, both co-purify together the complex which are in Peabody's and the complex which are outside of Peabody's. So we turned to a different approach, which was to engineer cells to get fluorescent Peabody's through the expression of one of the components, LSM14A fused to GFP here. We lyse the cells and we pass this lysate in a fax, in flow cytometry. We could observe a fluorescent signal that was specific of the Peabody's by comparison with cells which express a fluorescent protein as well, but not contained in Peabody's. So this signal here is specific from Peabody's. We sorted, using the fax, we sorted these fluorescent granules. After sorting, we spin them down reanalyze them on the fax, and they kept the same size and same fluorescence. So obviously they could resist the process that we call fluorescence activated particle sorting. Before sorting, it's very dirty lysate, with in green the Peabody's, and in red it's just aspecific staining of all membranous contaminants. After sorting, it looks rather clean. Then the difficulty was to purify enough material to be able to identify proteins by mass spec and RNA by RNA-seq. So we did quite an extensive purification, a 6 full day of sorting to purify 450 nanogram of protein that we could analyze by mass spectrometry. Here we compare the score, the mass spec score before sorting to after sorting. So we reason in terms of enrichment in Peabody's. In yellow are about 125 proteins which are enriched in Peabody's. In red are all the ones which were already known to be Peabody components. So they are mostly enriched. In black are a few proteins that we verified by immunofluorescence. They are indeed in Peabody's. But there are also many proteins which are significantly excluded. If we look at them, looking at the distribution of the proteins by function in the enriched protein compared to depleted protein, we got rid of most mitochondrial protein in violet here, cytoskeleton protein in black. We kept some motors, but we think it's relevant. It's related probably to the trafficking, the known trafficking of Peabody in the cells. And even in terms of proteins related, so 3/4 of the proteins are obviously related to nucleic acid metabolism. Even these have a specificity, like we got rid of all translation initiation factors in light blue, as expected from a granule which is not translating. We got rid of all stress granules markers in light blue as well here. So I don't want to insist too much on this protein content because we had enough material to do one mass spec analysis, so it's not very quantitative or robust, but it was enough to claim that this purification was quite to a sufficient extent to analyze RNA.
Can I ask a question?
Yeah, of course, of course.
This is Deanna. That's really beautiful. I was wondering how many cells did you need to extract?
300 millions. 300 millions.
Okay, great, thanks.
Yeah, so probably the yield is not very good because by calculation, starting with 300 million cells, we should have got more Peabody than what we really got. So in particular here, maybe there are still Peabody, like small Peabodys here that we missed, we don't know. Or maybe some still dissolved during the fax, we can't know, but at least the material we get at the end is enough to do the mass spec. But the yield is probably not very good. So in terms of RNA, this time we had enough material to 90 nanogram of RNA. So this was enough to do a triplicate RNA-seq, independent sorting and independent libraries. So in red is a profile in size, the size profile of the RNA from the purified Peabodys compared to the profile before the sorting in green. So it's not a few RNA species, obviously. It's a large range of size. The RNA are very much depleted in the 2 peaks for the ribosomal RNA, as expected. And in fact, it's a very, very large number of RNA which are significantly enriched. It's more than 6,000 of them. But there is also a very large number of RNA which are significantly excluded, more than 7,000. If we look at what they are, close to 90%, 89% of the RNA which are significantly enriched correspond to messenger RNA, while the long non-coding here in light blue or the antisense in blue, they are mostly excluded from the Peabodys. this is both coding and non-coding. If we consider the coding transcriptome, it's 1/3 of the coding transcriptomes, which is found enriched in Peabody's. So a very, very large amount or proportion of the coding transcriptome. I have no time to discuss that here, but it's published. We couldn't find any sign of 5' decay in the reads from the analysis of the reads of the enriched RNA. So now we compare this data set to what happens when we disrupt Peabody's through the silencing of one of the, always the DDX6 protein, proteins completely required for assembling Peabody. So we are going to compare cells, the transcriptome of cells which have Peabody and cells which do not have Peabody's. And we did a polysome profiling. So when looking at the total mRNA, this should reveal any function in mRNA stability. Here we compared what in the y-axis, what happens in terms of abundance of total mRNA, and on the x-axis, it's enrichment in Peabody's. The RNA which are found in Peabody's, they are not increased or decreased when we dissolve Peabody's. So they were not degraded in Peabody's. They don't mind the fact in terms of abundance, they are not sensitive to the presence or absence of Peabody's. However, when we look at the polysomal RNA, and here it's a polysomal to total ratio that should reflect any regulation of translation, we find that the RNA which are in Peabody's, they are more translated when we disrupt Peabody's than before, which is an argument to think that the Peabody's are a site of storage that prevent translation. So no increased stability, because it has been postulated before that Peabody's would be for decay. This was mostly coming from yeast studies, but there were also a few studies in human cells. We could not really find any argument for a role in stability of mRNA. This is pointing to translation, a function in translation repression. Now we could combine all these data, we could cross them with all the many existing CLIP experiments against RNA binding proteins, which identifies the targets of these RNA binding proteins. So I will make it short because it's a complex slide, but here you have the set of proteins, of RNA binding proteins against which the CLIP were performed. We rank them depending on the accumulation on the y-axis of their target in Peabody's. So for instance, on the left, FXR1, its targets identified by CLIP are massively enriched in Peabody's. On the other side, Ataxin-2, its targets are massively excluded from Peabody's. And then it's just ranked depending on the accumulation. In red, you have here all the proteins which we found in our mass spec or other groups found being present in Peabody's. And underlined in green, it's the one which were shown to be absent from Peabody's, particularly Ataxin-2. It's particularly true for Ataxin-2. So as you see, the proteins which accumulate in Peabody's, they tend to accumulate with their target RNA. These red proteins, many of them, they are known as translation regulators. So this is a second argument to think that the Peabody's are a place of storage for mRNA which are repressed at the level of translation.
Excuse me, I have a quick question.
So, of course.
You mentioned the DDX6 was 170-fold increased in Peabody, but the total number of protein is still below 20% in total.
Yeah.
Right, it's enriched. So is this mRNA enrichment kind of expressed the same way? There are a lot in the cytosol as well, or it was just? So
we can't really, this is a calculation we cannot do because for the first 170-fold enrichment of the DDX6 protein, this is a conservative analysis. Like we just look at section of cells, and in this section, we count the density of gold particles inside and outside. So we are sure not to miss anything. But here
Okay.
we are going to compare a sample before FAPS and after FAPS, and we have no idea of the yield. So we can't really come back to the same type of, we can discuss in terms of enrichment, but we can't discuss in terms of absolute ratio.
Right. Thank you.
Yeah. So our conclusion from this data and other added in detail is that human P-bodies are primarily involved in mRNA storage and not so much in mRNA decay as had been proposed earlier. Then we also considered what is the protein yield of the mRNA depending on their localization in or out of P-bodies. So I assume that you know that the protein yield, so what I call protein yield is what is the abundance of a protein with respect to the abundance of its cognate mRNA. Everyone knows, I think, that it's not really very much correlated. There are obvious reasons, like if a protein is very unstable, of course, it will give protein yield, which is lower than if the protein is particularly stable. But another source of protein yield variation can also be translation regulations. So here I plotted the protein yield for all the transcriptome, and this is taken from the literature for the quantitative protein levels and mRNA levels from our own data. And we just color-coded depending on the localization of the mRNA in P-bodies in yellow or outside of P-bodies in blue. And as you see, the protein yield is lower for the RNA which are in P-bodies compared to the one which are excluded from P-bodies. But even more, the correlation coefficient between the amount of protein and the amount of RNA is also, it's more correlated for the one which are excluded than for the one which are enriched in P-bodies, which is very much suggesting a regulation at the level of the translation. So I ask you to remind me about this protein yield story because I'm going to skip to something else, but I will come back to it later on in the seminar. So now, which type of mRNA are stored in P-bodies? When we look at the gene ontology categories, which are in, which are out, so it's a large variety because, as I said, it's 1/3 of the coding transcriptome, so it concerns all types of a very large panel of function. But still, there seems to be one constant, which is that the RNA which are in P-bodies, they tend to encode regulating function at the level of chromatin, transcription, RBP, PTM, but also many other ones, even for mitochondria or other type of function. While the RNA which are significantly excluded, they tend to encode basal functions. To be more explicit on that, I am going to drive you through a few examples. For instance, if I take protein degradation, it's operated by the proteasome. Here is the enrichment on the y-axis of the in P-bodies of the mRNA encoding. Here the different subunits of the proteasomes. Most of these mRNA, they are excluded from P-bodies. However, the RNA which encodes the protein which will regulate the targeting of proteins to the proteasome, namely the E3 ligase or deubiquitinase, they mRNA tend to be enriched in P-bodies. Its tendency is not systematic, but it is a tendency that we found in many different processes. If you think to RNA processing, so the splicing is done by these NERPs, the messenger are excluded, but the splicing regulators here, they tend to be enriched. LSM128 is also a constant, like an heptamer that control splicing, and it's also excluded. Yes. If I looked at mRNA translation, the ribosomal proteins, they messenger are massively excluded, whether belonging to the cytoplasmic, cytosolic ribosomes or mitochondrial ribosome, they are excluded from P-bodies. But regulator of translation like proteins of the CPB complex or protein of the RISC complex, their messenger RNA are enriched. And there would be many further examples. I took the ones which are the most familiar to me. So it suggests that this Peabody-related regulation may be involved in producing the right protein at the right moment, so the right regulators when required by the cells. And even more in detail, it seems that proteins that are going to interact together, their messenger are co-regulated. It's obviously, so either co-excluded or co-enriched in Peabody's. It's obviously the case of the proteasome, a big complex, or the SNRP, or the ribosome, or this LSM128 heptamer. But it's also or the CPEB complex or RISC complex, whose messenger RNA are enriched in Peabody's. This raises a possibility that the regulation associated to Peabody localization could also contribute to the protein stoichiometry in complex. It's a speculation, but we found it interesting. So now, if I just summarize what I presented to you, so in Peabody's, we find 1/3 of the coding transcriptome. These mRNA are rather abundant, I didn't say it, but they are rather very well expressed. However, they have a very low protein yield. The storage is reversible. This is from our polysome profiling experiment, but it has also been shown by others. 1/3 of the coding transcriptome is significantly excluded. These mRNA can be also very abundant or not, more or less, but they have a high protein yield. And all these RNA are decayed outside of Peabody's. Just of note, decay has also not been seen in Peabody's using RNA decay reporters by a series of groups. So I think in human cells, at least, it became a consensus that the decay is occurring outside and Peabody's are for storage. And in terms of function, storage is rather for mRNA encoding regulatory proteins, while the mRNA encoding basic function are rather excluded. So now I reconsider this story about protein yield. It could be, at first glance, it could seem very logical that a granule which is for storage contains mRNA very inefficiently translated, have a low protein yield. But the main question is which fraction of a given mRNA can really be stored in Peabody's, because I also insisted that the Peabody are very small in size and they are very few per cell. So we address this question using single molecule FISH. So here I show the data for the SPEN mRNA, which is an mRNA which is tenfold enriched in our purified Peabody's, which is quite a high level of enrichment. When we do the single molecule FISH, so here in red, each dot is a single molecule of this RNA. Yes, we see, if we look at that enlargement, we see clusters of this mRNA in Peabody's, here in green. But we see also many other molecules outside of Peabody's. So when we quantified that on a cell basis, so we counted the number of molecules in and out Peabody for 40 cells here. We found that it was very heterogeneous between cells, but in average, it was not more than 15% of the molecules of SPEN mRNA that were located in Peabody's, which was much less than what I would have expected. So we have to consider that exactly like the proteins, the mRNA, they can be enriched in Peabody's, but nevertheless, they always exist outside of Peabody's. And maybe for people interested in LLPS, it's just very logical because the phase transition is just a new balance between the condensed and a soluble phase, which means that it could have been expected that both the protein and mRNA component would be present in the dilute phase, even if they are very much concentrated in the condensed phase. Nevertheless, we have a problem in terms of amount because 15% of spent mRNA molecule in Peabody is never going to explain how this mRNA have 100-fold lower protein yield than most Peabody excluded mRNA. So in other words, it's not the sequestration of this mRNA in Peabody's that can explain this very low protein yield. And we had to conclude that this low protein yield is an intrinsic property of the Peabody mRNA. It's not related, it's not due to the sequestration in Peabody's. So our next task was trying to understand what could be the reason for this very low protein yield. We first looked at mRNA length because Parker's group on stress granules had noted that the RNA which accumulate in stress granules are longer than average. So here we divided the human transcriptome in 6 classes depending on the mRNA length, from less than 1.5 kb to more than 10 kb. And like in stress granules, the longer RNA accumulate more in Peabody's than the shorter ones. The correlation coefficient is 0.39, so it's relatively high or moderately high. However, what was the most striking feature of this RNA is the GC content. Here it's subdivided depending on the GC content. Transcriptome is subdivided depending on GC content from less than 40% GC to more than 60% GC. And obviously, the RNA which have a low GC content accumulate much more in Peabody's than the one which have a high GC content. And this time the correlation coefficient is minus 0.63, so very high. Can be represented a different way. Here it's a distribution of GC content in gray for the whole transcriptome from 35% to 70%. So for the human transcriptome. It's like a bimodal distribution, and only the AU-rich RNA accumulate in Peabody's here in red. The ones which are DC-rich, they are completely excluded here in blue. So it's a very striking effect of the GC content. It's related to, so here's a representation of the GC content over the length of the whole RNA, but if we try to distinguish the contribution of the GC content of the 5' UTR, CDS, and 3' UTR, we find that it is related to the GC content of the CDS and the 3' UTR, not so much of the 5' UTR. So if it's related to the, if there is a very strong bias of the GC content in the CDS, that should have it's expected to have a strong effect on the coding property of this mRNA. So we therefore looked at the amino acid usage in the CDS of mRNA, which are in red or out in blue Peabody's. For the 20 amino acids here, as you see, if I pick up a few examples, taking alanine, alanine is much less frequent in the, it's much less present in the protein encoded by Peabody mRNA than encoded by the mRNA which are excluded from Peabody's. It's the same for glycine. And in reversely, the lysine or asparagine, they are much more frequently encoded by the mRNA which are in Peabody's than the one which are outside. So this very strong GC content bias has consequences on the amino acid frequency in the proteins encoded by Peabody mRNA. I have no idea which type of property, I could not make a coherent picture of which type of property it would lead to, but it's an observation. Beyond this amino acid usage, it should also have an effect on codon usage. So here I represented for the 20 amino acids, it's a ratio of usage between the mRNA which are in Peabody's and the one which are out. So take the leucine, for instance, it is encoded by 6 codons. one codon here is twofold, it's a log 2, sorry, so fourfold more used in Peabody mRNA than in Peabody excluded mRNA. And of course, some are less used. In every single case, it's completely related to the percentage of GC in the codons. It's color-coded in gray here. So this codon, which is more used for leucine, is completely devoid. It's only AU, composed of A and U, and it's devoid of G and C. And the one which are less used, they have more GC. So the GC content bias leads to a codon usage bias in Peabody mRNA. And this is really interesting because in human, the codons which are less frequently used, most often they end with an A or a U. So this led us to think that maybe it was related to the low protein yield that we observed earlier. So we plotted the frequency of these low usage codons with the protein yield. As expected, we see that the RNA which are in Peabody's in red, they have a much higher frequency of low usage codons because they are just much more AU rich than the one which are outside of Peabody's. But the correlation with the protein yield is moderate, 0.2. However, I also told you that the mRNA which are in Peabody's, they tend to be longer. And this is also true of the CDS. So if we combine the two and this time plot the absolute number of low usage codons per CDS, this is still very correlated to localization in or out of Peabody's. And this time it has a good correlation with the protein yield with the correlation coefficient of minus 0.46. So we think that we have found what was the intrinsic properties of this Peabody mRNA to have a low protein yield. The reason is the GC content, which is very poor. Okay, now to extend that, we thought, okay, GC content, we were very surprised by that. So we did check and recheck several times, but there is no problem, it's true. So we were wondering if it's very important for Peabody localization, maybe it's also important for other aspects of post-transcriptional control. So we addressed first that question by considering very general factors involved in mRNA decay or translation. So I told you we have the polysome profiling after silencing DDX6. DDX6 is known as both partners of the decapping complex that stimulate the decapping in yeast, but also as a translation repressor. We generated transcriptomes after silencing XRN1, the exonuclease, which is responsible for the 5' decay, and after silencing PAT1B, another factor that is both known in yeast as a cofactor of decapping and in all species as a cofactor of translation repression. And I just give you a flavor of it through this heat map. So here we presented the whole transcriptome, and we clustered it depending on the GC content, Peabody localization, and behavior of the mRNA after silencing the DDX6, XRN1, or PAT1B. And we also have the dataset of polysomal RNA after silencing the DDX6. And as you see, just the hierarchical clustering of these genes lead to the separation of two main groups of RNA, which are more or less the AU-rich RNA and the GC-rich RNA. The GC-rich RNA, they decay, or the abundance, so they decay, is sensitive to the silencing of the exonuclease XRN1 and the helicase DDX6, but not of the silencing of PAT1B, which was not expected at all. We thought that PAT1B and DDX6 had exactly the same targets. And these RNA, they are excluded from Peabody's. And conversely, the RNA which are AU-rich, the one which are in Peabody's, they are very insensitive to silencing of XRN1 and DDX6 in terms of decay. However, the decay is controlled by the level of PAT1B and the translation by the level of DDX6. So now, if it's important for this, so first, sorry, these general factors that we thought were general at the beginning of the study, they are not so general. They have a large number of targets, but they have preferences for either GC-rich or AU-rich target. If it's true for these general factors, is that also true for the sequence-specific factors, sequence-specific RBPs? So we took a set of factors and use either clip data or search for identification of mRNA binding motif. And we observed that, in fact, the targets of all these RBPs, they have bias in terms of GC content. Some of them, here's the median in red. Some of them have targets which are more GC-rich. Some of them have targets which are more AU-rich. And here in this heat map, it's just ranked depending on the GC content. As you see at the top are the two only factors we could find that were clearly decay regulators and only decay regulators. YTHDF2 is related to M6A dependent decay and SMEC6 to the nonsense mediated decay. And the other one here at the bottom, so liking AU-rich RNA, most of them are translation regulators. And if we look at our other data sets, the behavior in the different data sets, including the p-body localization, is mostly expectable, can be predicted from the GC content. I would have thought that being a target of Pumilio, for instance, would make a huge difference with being a target of HUR. But it's not the case. They tend to follow exactly the behavior of RNA of similar GC content. There are a few exceptions. I have no time to detail, but in general. So we wondered what was the situation for mRNA targets. And we took the targets of the 22 mRNA, which are the most expressed in our cells. And it's very similar. In most cases, the targets of these microRNA are AU-rich, except for these two ones. In most cases, they are A-rich. And their behavior mostly follows the GC content, maybe with more variation. But particularly the accumulation in p-bodies is completely related to the GC content, with very few exceptions. So it seems that the mRNA-specific, sequence-specific factors, the decay regulators, they target are GC-rich. The translation regulators, they target are AU-rich, including the mRNA target. So now this is for the CDS. So our next question was, I told you that the GC content was important both at the level of the CDS and at the level of supramutea, or at least in terms of correlation coefficient. We could not make the difference in our first analysis. So our question was, okay, the CDS, it has an impact on mRNA regulation, mRNA control, but what is the function of the supramutea on the top? Does it have function, or just because the CDS the GC content of the CDS and the GC content of the supramutea, they are very highly correlated together. So can we distinguish the function of the two? So it's what we try to do in this type of analysis, where we consider mRNA depending on the GC content of the supramutea, and for each bean, we look if the GC content of the CDS makes a difference. So in all cases, whatever is the GC content of the supramutea, in every case, the GC content of the CDS is lower if the RNA are in than if the RNA are out. So this is a primary factor, that the GC content of the CDS is much lower for mRNA which are in p-bodies. And we think it's related to translation efficiency. In fact, we can also look at link RNA, which cannot be translated. They are mostly excluded from p-bodies, and the correlation is very poor with the GC content. It's -0.2, while it was -0.63 for mRNA, for coding messenger RNA. Can we show it directly in a cell experimentally? So we use reporter plasmids, so luciferase reporter plasmid that we transfected in hex cells, except that we just switch the coding sequence, a GC-rich coding sequence, to AU-rich coding sequence. So they encode exactly the same protein, it's just a codon usage which is different because we change the GC content. We transfect them in hex cells, and then in the cells we visualize the P-bodies in green, and we visualize the messenger RNA by single molecule fish in red. As you see, this, if we have a GC-rich CDS, it does not accumulate in P-bodies. There are very few molecules. There are no clusters of RNA molecule in P-bodies. However, with our AU-rich CDS reporter, then we observe clusters of RNA molecules in P-bodies. So changing just the CDS GC content is enough to change the localization with respect to P-bodies. And this was quantified here. So in two independent experiments, we have much more clusters of molecules in P-bodies if the reporter is if the CDS is AU-rich than if the CDS is GC-rich. Of course, that has also an impact on protein yield. An AU-rich CDS provides more protein than a GC-rich CDS, despite the same amount of RNA. Now, okay, for CDS, there is an impact of the GC content. If we want to show if there is also an impact of the 3' UTR, we have to do exactly the reverse analysis. So we fix the GC content in the CDS and look if the GC content in the 3' UTR is dependent in and out of P-bodies. If the CDS is AU-rich, we can't visualize any contribution, any particular contribution of the GC content in the 3' UTR. However, if the CDS is GC-rich, then the RNA which accumulate in P-bodies, they have a GC content in the CDS, in the 3' UTR, sorry, which is much lower than if they are excluded. So there is clearly also a contribution of the GC content of the 3' UTR, at least when the CDS is GC-rich. And we postulate that it could be through the capacity of this 3' UTR to bind factors that like AU-rich sequence and favor condensation into P-bodies. Again, can we give some experimental argument? For that, we set up an in vitro constitution assay on eyes. So we have our cells with fluorescent P-bodies. We lyse the cells, eliminate the nuclei, eliminate the P-bodies, the pelleted P-bodies here under microscope, they look like that. And then we have the cytosol, the supernatant, which is devoid of P-body, but still contains some soluble LSM14A, which is fused to GFP. And we add to this cytosol purified recombinant DDX6 protein. That reforms granules that look like that under microscope. So they are very similar to the pelleted P-bodies. We can count them by FACS. The formation is DDX6 dose dependent. And then on the top, we can add RNA. So either RNA sensitized in vitro from the GC-rich reporter or from the AU-rich reporter. When we add on the top GC-rich RNA, then we tend to form less granules here that we call P-body-like granules. Just we can't claim that they are P-bodies, but at least they contain LSM14A and DDX6. So if we add GC-rich, that prevents the formation of granules. However, if we add AU-rich RNA, that favors the formation of granules. In that case, it's on eyes. The RNA, they are not polyadenylated, they are not capped. So we think this reflects the contribution of the RNA as such, independently of translation, to favor condensation of P-like granule. So if I summarize all that, in human, there are large variation of GC content in the transcriptome. The RNA, which are GC rich, they are very highly translated, very efficiently translated. They bind a set of specific factors, mostly related to mRNA decay pathways. They are decayed through a pathway involving the 5' exonuclease XRN1 with the help of the DX6 helicase. It's a helicase, so it can be useful to unfold a GC-rich RNA. While the ones which are AU-rich, they are poorly translated. They bind a bunch of other factors that like AU-rich motif. They decayed is depending on PAT1B. We speculate that it's from the 3', but we have no direct evidence of that. The translation repression depends on the DX6, and they are the ones which accumulate in P bodies. This effect of the GC content is due to the GC content of the CDS, probably through translation efficiency, but also to some extent to the GC content of the 3' UTR, and maybe through its factor binding capacity. And lastly, I just want to add this comment. So I was thinking that this type of model could be particularly relevant for haploinsufficient genes, which are genes which by definition have a limited capacity of producing proteins. So there are some papers that gave over genome-widely some probability of being haploinsufficient. So I used that, and here is a representation that the RNA which are in P bodies in red, that they tend to have a much higher proportion of genes with high probability to be haploinsufficient than the one which are excluded from P bodies. So probably that many of these regulations apply to the RNA which encode, which come from haploinsufficient genes. And so to conclude, just the people who have been involved in this work. So these are the people of my group, particularly Maïté Courel, who was involved in the polysome profiling experiment, Michel Hernoulange, Marianne Bédard in the immunofluorescence data, Michel Kress in some bioinformatic analysis, Arnaud Hübchen-Berger, who was the first actor of the P-body purification, who now has his own group in Nice, Rasha Chouaib, who initiated the single fish molecule experiment. And then we had plenty of collaboration with platforms, but also for electron microscopy with Gérard Piron's group, for the various data sets after silencing from Nancy Standard's group, Patrick Brest's group, Antonin Morieux's group, and for codon usage analysis, the Hugues Röstrolius group. And I thank you for your attention.
Thank you very much. You've covered so much. Very interesting.
Maybe too much. Maybe too much.
No, it's great. It's phenomenal. I mean, I could ask a lot of questions, but I'm sure many other people have questions too. I'll just ask one. I'm really interested in the idea that different types of condensates may use different helicases.
Mm-hmm.
You know, so your particular work on DDX6, you know, how much is that sort of teaching us a more generalizable sort of lesson?
So I don't. Yeah. My first answer would be that there are many answers that can be done. First of all, in our mass spec analysis of the purified P-bodies, we noted that there is an overrepresentation of helicases. So we didn't test all of them. We tested DDX3, DDX3. DDX3, we silence it. This is not required for making P-bodies. So there is another representation of helicase in there, but it's very particular to DDX6 to be required for assembling P-bodies. This is one thing. There may be some others. We didn't try. I mean, there are many, many different DDX in the transcriptome, and we haven't had time or money or energy or hands to test all of them. But second, it cannot be completely universal, like even for DDX6 in yeast. DDX6 is not completely required for making P-bodies. Yeast P-bodies, they seem to be different. And you have to silence both DDX6 and PAT1B to observe the disappearance of P-bodies. So even the function of P-bodies, of DDX6 for P-bodies, I always insist that it is in human cells, but because in yeast, it seems to be different.
Interesting, thank you. Other questions?
Hi, yeah, I have a question about the FAPS purification. So I'm wondering if in your RNA extraction, you're able to capture microRNAs and whether the microRNAs are depleted or enriched in your P-bodies.
This is a very interesting issue, but unfortunately, what we purified was just a standard RNA purification protocol, so that we didn't purify the microRNA, and the library particularly we prepared, they were for over 200 nucleotides. So we have no idea about microRNA. Some groups have shown by in situ hybridization that microRNA can be present in P-bodies one by one. But we have no data. It would require for us to redo a complete purification and have enough material to do microRNA preparation and a specific library to this to answer this issue. So we don't know. We can only look at the targets.
Hey, Dominique, super interesting talk. Thank you very much. It's beautiful work. I was wondering on the also on the FAPS method you use to purify the P-bodies. I found it very interesting that the P-bodies are actually stable in the cell lysate. Have you ever investigated the material properties of the P-bodies in the cell and in the lysate? Does that change? Is it more gel-like? So how do you explain the stability? It's very interesting.
So yeah, the stability, I don't explain anything. Many people said, well, how is it possible to purify them by FAPS? Because FAPS, our procedure is more or less an extreme dilution, is a purification by dilution. So one would expect that they should dissolve, and they didn't dissolve. Or I insisted that the yield is low, is poor. So maybe some dissolve, but at least the material we have, we can spin it down and then put it back in the FAPS, and we still have material. So they seem to be very resistant to dilution. I don't know. In the FAPS procedure, there is no cross-linking, there is no particular treatment, so there is no trick, and it's they remain more or less intact. I can't say intact, but we found all the protein expected to be there. They were there, so they. The only thing I can speculate is, from what I discussed with biophysician, it's not the concentration dependent has not to be symmetrical between condensation and dissolution. So there may be a certain concentration, after a certain threshold, it condensates. But for dissolution that it would require, for instance, ATP. I say ATP because there have been quite a few papers saying that maybe ATP and helicase are important for condensation. And somehow we got completely rid of ATP by our extreme dilution as well. So maybe, and we didn't try that, if we would add back ATP, maybe they would dissolve. I don't know, there has to be something that disappeared in the surrounding cytosol that make them so resistant, or at least not completely concentration dependent. This is one thing.
Yeah, that's super interesting. And it also loops back to Mark's question, right? So ATPase driven machines like helicases or maybe chaperones like HSP70 has been shown to be important for dynamics of stress granules. Maybe it's conceivable that these machines are also important for keeping up the dynamics of P-bodies, and would be very interesting to study these specifics in cells.
There's Carsten Weiss in Switzerland has performed experiment on the DDX6 in yeast. And in yeast, he's saying that when he's mutating the helicase function, ATPase activity of the DDX6, then these stabilized P-bodies, they are more stable. in human, we did this type of experiment a long time ago. I think we published that in 2008, so 12 years ago now. We performed several mutations of the DDX6 helicase and did complementation assays. None of them was able to assemble PBDs, meaning we silenced the endogenous DDX6 protein, and then we transfect the cells with construction to express various mutants of DDX6. This was based on the mutation performed for other DDX6 helicase in other systems. None of them was able to reassemble PBDs, while this complementation assay is working very well. If we use a wild type protein, it works very well. So in human, the helicase function is required for condensation. We don't know if it's for condensation in terms of local condensation or some upstream regulation even, which is required for the RNA to be able to condensate together, but it is required. And it seems to not to be the case in yeast. And Carsten Weiss has written a long review on the function of the helicase, but his observations are very restricted to yeast, at least for DDX6. So this very large enthusiasm about the function of helicase, yes, there is a function of helicase, it's in helicase, but I don't know how to make generality of it, because the most generalized review or paper that was published recently does apply to yeast DDX6, but not at all to human DDX6. And now just to complete, so you said that, do you know anything about the properties? This is a very small object. Like, it's not like stress granules. Stress granules, you can do FRAP experiment very easily on them. PBDs, they are less than 500 nanometers diameter. And personally, I was not successful in frapping them. Most of the case, they would just flow away, and I would just lose them from the focus. There are a few papers who described some FRAP experiment on it, and they say it's very common, like a half a minute recovery, or sometimes a little bit less, sometimes a little bit more, but it is in the range observed for others. But doing really accurate measurement of fusion time or things like that, I don't think there is anyone who was successful for that.
Hi, Dominique, this is Violetta.
Yeah.
Thank you so much for being here. I'm just so excited that you gave this seminar, and it's just fabulous work. I have a few questions. You mentioned about reversible storage of mRNAs and P-bodies. Is there anything known about those mechanisms and what that is in response to in terms of like signaling pathways?
No, not really. There are not many example of natural situation where suddenly the PBD disappear, except in mitosis. In mitosis, like all other condensate, the PBD completely disappear. Nevertheless, we got interested. I tried to cross our data with data of translation regulation during mitosis to look if there would be some particular increase of translation of the RNA which are found in PBDs, and it's not the case. So it's not because they are obviously released from PBDs, because PBDs dissolve at mitosis, but the translation doesn't resume, or at least not enough for people to have noted it when they look at mitosis-specific translation. So, and beside that, I'm not aware of any situation that would drastically change the number of PBDs. However, what I can imagine is that, what I imagine in fact, if that's for regulation, then it's not because the object seems to be maintained all the time, but its content is maintained all the time. And what I imagine is that the content is renewing all the time, and depending on the trigger, depending on the environment, on the cell culture, whatever, hormones. But the object as such, the PBD as such, doesn't look changed. I don't know if I'm very clear in what I'm saying.
Yeah. That the content,
like the RNA have to go out and in, but selectively, or it would not make sense when I give the landscape where inside its mRNA that tend to encode for regulators. The cell doesn't need all regulators at once. It needs the regulator one after the other. So I imagine that the content changed, and from time to time, the cell needs some type of regulator, but not others. But globally, at middle scale, we don't see any difference in terms of size of P-body or morphology of P-body.
Yeah. I have two more questions. I think you started to touch upon, you talk about the haploinsufficiency, but I was interested to hear about any diseases that where P-body dysregulation is implicated. I guess it's too early to know.
Yeah. It's just an observation that these haploinsufficient genes, which are haploinsufficient, so just one version of the gene is not enough to produce enough protein. You need two of them. So it means that they have a limited capacity of producing protein, and they correspond to the one which are in P-bodies. I thought it was interesting, even if I can't apply it to anything particular.
Yeah. And then the other question, Alicia was kind of pointing out. So, you covered that the P-bodies contain mRNA targets, microRNA targets. I guess it's difficult to look at mRNAs themselves. I'm curious about mRNAs a lot, just because there's a lot of preclinical data and some clinical data now emerging in regards to using mRNAs as therapies. And so it makes me curious about its function in the P-body, in particular because a lot of its targets are in there, and whether or not efficiency of these therapies may depend on its ability to get into the P-body. Another question I have is, if mRNAs are stored in P-bodies, if they're enriched in the P-bodies, perhaps there's a way to manipulate, the in and out of the mRNAs, so that cells become their own source.
Okay. So manipulating the in and outs, this seems very far. I don't know. I have some difficulty to envision it. However, if you would like an RNA to go into P-bodies, definitely I would advise first to try an AU-rich one. I would bet that the GC-rich one is not going to work. So I would try to put at least a large part of the RNA AU-rich to make sure it goes in. Now, to control the in and out, we don't know enough. Like, we wouldn't first need to know why it's going in, which is it depending on one single protein. So on the whole data set, I didn't show it, but we tried to, at the beginning, we were dreaming, particularly the first author of the paper.
Dominique is frozen.
Yeah, frozen for me
Paper
too.
was dreaming that maybe very. Okay, that's fine.
It's better now. Thanks.
Well, I'm sorry. I think it's my connection. But... So yeah, then I repeat. At the beginning, I was hoping, and the first author of the paper was also very much hoping that there would be like a zip code, like some particular motif related to some particular protein that would be enough to drive RNA in P-bodies. So we did a PCA analysis of the whole data set, and we found that, yes, there are proteins that are, protein binding is related to the fact that it can accumulate in P-bodies, but there is no single combination. So we didn't go further to analyze it in detail, but it's not, there are probably many different factors and many combinations of them that can drive an RNA in P-bodies. So as long as we don't master that, I can't imagine how we could master the in and out trafficking.
Thank you.
Hi, Dominique.
Hi.
I have a question around the changes in the translation of mRNAs based on being in P-bodies. So I might have missed this, but when you guys dissolved P-bodies by knocking down DDX6, did you measure translation differences there? And is that where you saw the differences?
Yeah, it's where we did it was a polysome profiling.
Okay.
So then we measured we quantify the RNA with total RNA and polysomal RNA. And we say when we dissolve, then we see that the RNA which were before in Peabody, they tend to be more translated. It's a very limited extent, but which can it's like 30% increase. It's not a huge increase, but it's consistent with the amount that can be in Peabody's.
Yeah, because I was gonna say it seems like that potentially isn't super consistent with the fact that, because you also found only like 15% of the RNA is in the Peabody to begin with. So if that's the case, you could only ever expect a 15% increase, right? So I guess where the increases on that magnitude, I can't remember.
So the extent of increase in polysomal RNA was consistent with the amount of RNA that is not sequestered, but in Peabody's at a certain moment.
Okay.
And this is not enough to make a huge regulation.
Okay, cool. Thank you.
So it is reversible, but it's not a factor 10. It's like a very small factor.
Yeah. So given that, I guess, do you think that's the primary function of Peabody's then, like very fine-tuned translational regulation, or is there any other kind of?
I would think it's completely it's completely open. Either it's really fine-tuning, and maybe in the past, I don't know, in the not very old past, where people were saying if it's not a factor 2 difference, it's not biologically relevant. I think this is wrong. So sometimes 20% difference make a difference. So then it would have to be genes that are very sensitive to very small differences. Or another possibility is that what we see is only the condensation. Maybe something happened in the condensate, but then the RNA is released, but still has been marked for something that is going to impact its fate. Then we still should see it when we do our silencing experiment to look at cells after dissolving the Peabodys, and we don't see evidence of that, but maybe we don't look at the right thing, like we only look at translation and decay, at abundance of RNA. Maybe we should look at modifications of RNA, or maybe we should look at, I don't know, the exact cap. What is the definition? What is the exact identity of the cap? Maybe something could change when it goes through the condensate. That's one thing. Or alternatively, maybe we don't ask the right question at all. And what is important is not the amount in the condensate, but rather that the condensate can buffer some differences in the cytosol, in which case we just focus the wrong way.
Okay, thank you.
That's a subtle topic. Yeah. Other questions? Well, if not, I think we're close to time anyway. So Dominique, we just want to thank you again. Great lecture, and very much enjoyed it.
Okay.
Okay, and we may have some other follow-up questions we'll come back to you with, if that's okay.
Of course.
Yeah. You know how it is. Sometimes things hit you later.
Yeah, no problem.
Really appreciate it.
It was nice to do my presentation. A little bit strange, but
it is. It is a little weird, but it's still great to have the chance to connect like this. So thanks very much.
Yeah.
All right.
Okay.
Take care. Okay. Bye-bye.
Bye.
Thank you.